Welcome to our microlearning channel. In today's video, we will be sharing on plants, focusing on the model plan. Arabidopsis thaliana is a small flowering plant that serves as a model organism for plant biology research. Its genome has been fully sequenced, and there are many genetic tools available to study this plant. The root system is essential for the plant's growth and development. Roots absorb water and nutrients from the soil, anchoring the plant in place. This slide provides an overview of the importance of the Arabidopsis root system. Arabidopsis is a small flowering plant that has become a crucial model organism in plant biology research. Its small genome and quick life cycle make it easy to study and manipulate genetically. There is a wealth of research resources available for Arabidopsis, including mutant libraries, databases, and well-established protocols. Insights gained from Arabidopsis research often apply to other plant species, including important crop plants, due to shared genetic pathways. The global community of Arabidopsis researchers is large and highly collaborative, driving rapid progress in our understanding of plant biology. The Arabidopsis root is a fascinating structure, with several key components that work together to support the plant's growth and development. The root cap at the tip protects the delicate root apical meristem as it pushes through the soil. The vascular tissue, made up of xylem and phloem, is responsible for transporting water and nutrients throughout the plant. The epidermis is the outermost layer, and the root hairs that extend from it greatly increase the surface area for nutrient absorption. The cortex stores food and water, while the endodermis regulates the movement of these resources into the vascular tissue. Understanding the anatomy of the Arabidopsis root is crucial for studying plant physiology and improving agricultural practices. The root apical meristem is a critical region at the tip of the plant root that drives primary root growth and development. This meristem contains actively dividing cells that give rise to all the major tissues and cell types found in the root. The root cap, epidermis, cortex, endodermis, pericycle, and vascular tissue all originate from the cells in this meristematic region. The image shows a close-up microscopic view of the root apical meristem, highlighting the vibrant colors and complex organization of the actively dividing cells. This region is essential for the plant's ability to anchor itself in the soil and absorb water and nutrients from the environment. The root cap is a crucial protective layer at the tip of the root. It shields the delicate root apical meristem as the root pushes through the soil. The root cap also helps the root sense gravity, allowing it to grow downwards. This image shows a close-up, detailed microscopic view of the root cap structure and cell layers. Understanding the function and anatomy of the root cap is important for plant growth and development. The epidermis is the outermost layer of the root, providing a protective barrier from the external environment while also facilitating the uptake of nutrients and water. The root hairs are specialized epidermal cells that extend from the root surface, dramatically increasing the overall surface area for absorption. Epidermal cells are tightly packed together and have a waxy cuticle coating. This cuticle helps prevent water loss and protects the root from pathogens and other threats. The cortex is a layer of parenchyma cells that lies between the epidermis and the endodermis in the root. These cortex cells are loosely packed and contain chloroplasts, which are involved in photosynthesis. The cortex is also responsible for storing and transporting water and nutrients throughout the root. The cortex plays an important role in root development, providing structural support and facilitating cell differentiation. This cross-section image shows the green chloroplasts within the cortex cells, as well as some of the nuclei. The endodermis is a single layer of cells surrounding the vascular tissue in the root. It plays a crucial role in regulating the movement of water and nutrients to the vascular tissue. The endodermis has a band of thickened cell walls called the Casparian strip. The Casparian strip prevents the apoplastic movement of water and nutrients. This ensures all water and nutrients must pass through the plasma membrane of the endodermal cells. This allows the endodermis to selectively uptake and transport water and nutrients. The pericycle is a single layer of cells just inside the endodermis in plant roots. It plays a critical role in lateral root formation, where new roots grow from existing ones. Cells in the pericycle divide and differentiate to form the vascular tissue of the lateral root. The pericycle also contributes to the vascular cambium, which allows the root to grow in diameter. This cross-section image shows the pericycle as a light green layer surrounding the darker green vascular tissue in an Arabidopsis root. The vascular tissue forms the central core of the plant's root system. It is composed of two key components, xylem and phloem. Xylem transports water and dissolved minerals upwards from the roots to the rest of the plant. Phloem transports sugars produced by photosynthesis downwards from the leaves to the roots and other parts of the plant. This integrated vascular system is critical for distributing essential resources throughout the entire plant. The detailed cross-section illustration helps visualize the structure and function of this important plant tissue. Root hairs are single-celled extensions from the epidermal cells of plant roots. They increase the root's surface area, which enhances the plant's ability to take up nutrients and water from the soil. Root hair morphology and distribution vary across plant species and can be influenced by environmental conditions. Root hairs develop in a specific zone near the root tip, a process influenced by hormonal cues and environmental signals. The primary root, which originates from the embryo's radicle, grows downward and establishes the initial root system. Lateral roots emerge from the pericycle of the primary root, increasing the surface area of the root system for more efficient water and nutrient absorption. Lateral root development can be influenced by environmental factors like nutrient availability and water stress, allowing the plant to adapt its root system accordingly. Understanding these key stages of root development is crucial for optimizing plant growth and productivity. Root gravitropism is the growth response of roots to gravity, with roots exhibiting positive gravitropism, meaning they grow downwards in response to gravity. The growth hormone auxin plays a crucial role in regulating gravitropism. Auxin accumulates on the lower side of the root, promoting cell elongation and downward growth. This helps ensure the root system can effectively anchor the plant and absorb water and nutrients from the soil. Roots have specialized cells that can detect water gradients in the soil, allowing them to sense areas of higher moisture. When these water-sensing cells detect a water gradient, the roots will curve and grow towards the water source. This directional growth, known as hydrotropism, helps the plant efficiently access and utilize water resources in the soil. Hydrotropism is an important adaptation that enables plants to thrive in diverse environments by optimizing their access to water. Root thigmotropism is a plant's growth response to touch or physical contact, a type of directional growth called tropism. When a root encounters a solid object, it senses the pressure and adjusts its growth direction through changes in cell elongation and division. This ability to navigate around obstacles in the soil helps the roots maximize access to important resources like water and nutrients. The image shows a close-up view of an Arabidopsis root growing around a small rock in the soil, illustrating this thigmotropic response. The root system plays a crucial role in nutrient uptake for the plant. Roots actively transport nutrients from the soil into the plant using specialized proteins. This process requires energy. Nutrients can also move passively from areas of high concentration to low concentration in the soil through diffusion. Root hairs increase the surface area of the root system, enhancing the plant's ability to absorb and uptake nutrients. Roots have mechanisms to selectively uptake certain nutrients, ensuring the plant receives the essential elements it needs. The root is the key entry point for water into the plant. Specialized cells in the root epidermis have a large surface area to absorb water from the soil. Water then travels upwards through the xylem vessels, which are long, hollow tubes that extend from the roots to the leaves. This upward water transport is driven by transpiration, the evaporation of water from the leaves. Transpiration creates a pull that draws water up from the roots through the xylem. Root respiration is the process by which roots, like all living organisms, obtain the energy they need to function. Roots break down carbohydrates like sugars through a process called respiration, which releases ADP, the energy currency of cells. This respiration occurs in the mitochondria of root cells, where oxygen is consumed to generate the ADP. Roots require a continuous supply of oxygen, which diffuses from the surrounding soil into the root cells. Oxygen is essential for this respiration process that powers the root's essential functions. Root exudates are chemical compounds released by plant roots into the surrounding soil. These exudates play a crucial role in shaping the root microbiome, They influence the composition and activity of microorganisms living in the soil. Some exudates can help mobilize nutrients, making them more accessible for the plant to take up. Root exudates can also act as defense mechanisms, deterring pathogens and attracting beneficial microbes to the plant's root system. The root microbiome is a diverse and complex community of microbes, including bacteria, fungi, and archaea, that live in and around plant roots. These microorganisms play a crucial role in plant health and development, forming beneficial symbiotic relationships with the plant. Many root microbes provide essential nutrients to the plant, enhance nutrient uptake, and protect the plant against pathogens. Root microbes also contribute to important nutrient cycling processes, such as nitrogen fixation and phosphorus solubilization, which are vital for plant growth and productivity. Some root microbes can even suppress plant diseases by producing antimicrobial compounds or competing with pathogens for resources. Understanding the diversity and functions of the root microbiome is key to developing sustainable agricultural practices that support plant health. Roots are the foundation of plants, and they face various biotic stresses that can significantly impact plant health and growth. One key biotic stress is nematodes, parasitic worms that can damage roots and stunt plant development. Some plants have evolved resistance mechanisms to combat nematode attacks. Insects like root-feeding weevils are another threat, as they can directly damage roots, leading to stunted growth and even plant death in severe cases. Pathogenic fungi can also infect roots, causing diseases like root rot that affect nutrient uptake and water transport, weakening the plant. Bacterial pathogens are another concern, as they can attack roots and cause diseases that further hinder the plant's ability to thrive. Understanding these biotic stresses and how plants respond to them is crucial for developing effective strategies to protect root health and ensure robust plant growth. Roots play a crucial role in helping plants adapt to abiotic stresses like drought and salinity. Under drought conditions, roots reduce their growth and increase root hair development to enhance water absorption and maintain water balance. Roots also enhance lateral root growth to explore more soil volume and find available water. High soil salinity creates osmotic stress for roots. To cope, roots accumulate osmolytes to maintain osmotic balance and adjust water uptake. Roots also have mechanisms to exclude sodium, which helps them survive in saline environments. Understanding these root adaptations is key to developing stress-tolerant crops. Plants have remarkable adaptability, and their roots are a key part of this. Roots can adapt to various environmental conditions to ensure the plant's survival and growth. Let's explore 4 key ways roots adapt to their environment. Water availability. Roots can develop deeper root systems or form lateral roots to access more water when it is scarce. This allows the plant to tap into water sources that may be deeper underground or spread out horizontally. Nutrient availability. In low nutrient conditions, roots can increase their surface area for nutrient uptake. They may also develop specialized structures to access specific nutrients more efficiently. This helps the plant obtain the nutrients it needs even in poor soil. Temperature fluctuations. Roots can tolerate temperature extremes by producing protective compounds. They can also adjust their metabolism to function in cold or hot conditions. This allows the plant to thrive even when temperatures fluctuate dramatically. Soil salinity. In high salinity soils, roots can regulate ion uptake or develop mechanisms to exclude excess salt. This prevents the plant from being damaged by the high salt levels in the environment. Roots are essential for plant growth and development, anchoring the plant and absorbing water and nutrients from the soil. The genetic regulation of root development is a complex process involving various mechanisms, including transcription factors, hormones, microRNAs, and epigenetic modifications. Transcription factors are proteins that regulate gene expression, controlling the synthesis of proteins that drive root growth and differentiation. Hormones, such as auxin and cytokinin, act as signaling molecules, influencing root development in different ways, like controlling growth and differentiation. MicroRNAs are small RNA molecules that regulate gene expression post-transcriptionally, adding another layer of complexity to the genetic control of root development. Epigenetic modifications like DNA methylation and histone modifications influence gene activity without altering the DNA sequence, contributing to the flexibility and adaptability of root development. Understanding these genetic mechanisms is crucial for improving root traits and optimizing plant performance in different environmental conditions. The root is a crucial part of the plant, responsible for anchoring it in the soil and absorbing water and nutrients. Understanding the molecular mechanisms behind root growth is key to optimizing plant health and productivity. Today, we'll explore the different processes that drive root development, from cell division to differentiation, and the various factors that regulate this growth. We'll start by looking at the root apical meristem, where new cells are constantly being added to the root tip. These newly formed cells then undergo rapid expansion, driven by water uptake and cell wall loosening, allowing the root to elongate. As the cells expand, they also begin to differentiate into specialized cell types, forming the distinct tissues of the root, like the cortex and vascular system. Plant hormones, such as auxin, gibberellin, and cytokinin, play a critical role in coordinating these processes, regulating cell division, expansion, and differentiation. External environmental cues, like gravity, light, and nutrient availability, also influence root growth patterns, guiding the root system towards favorable conditions. Underlying all of these processes are complex genetic networks, which fine-tune gene expression and protein activity to ensure the root can respond and adapt to its environment. Time-lapse microscopy allows us to observe root growth and development in real time, capturing dynamic processes that provide valuable insights into root biology. Various microscopy techniques, including light, confocal, and electron microscopy, give us high-resolution images of root structures, helping us identify different cell types and understand developmental processes. Controlled growth assays enable us to assess root growth and responses to various factors, allowing us to evaluate root architecture, branching patterns, and growth dynamics. Genetic approaches, such as mutant analysis and gene expression studies, provide crucial insights into the genetic pathways that regulate root development and function. The intricate root structure of Arabidopsis is beautifully captured in this high-quality microscopic image. We can clearly see the different tissues and cell types that make up the root, including the root cap, epidermis, cortex, vascular tissue, and endodermis. Advanced microscopy techniques like confocal, light sheet, and electron microscopy allow us to visualize these root structures in incredible detail. Understanding the anatomy of the root helps us better comprehend important physiological processes, such as nutrient and water uptake, as well as the plant's response to environmental stresses. This detailed visualization provides valuable insights that can inform our research into Arabidopsis root biology and development. Transcriptomics provides insights into gene expression patterns in root cells, revealing genes involved in root development, stress response, and nutrient uptake. Proteomics helps identify proteins involved in root growth, signaling pathways and interactions with the environment. Next-generation sequencing enables high-throughput transcriptome analysis, identifying thousands of genes involved in root function and revealing gene regulatory networks. Mass spectrometry is a powerful tool for identifying and quantifying proteins in root samples, providing information about protein modifications and interactions, crucial for understanding root function. The goal of mutant screening is to identify plants with altered root growth, morphology, or function by analyzing a large population for any variations. A random mutagenesis techniques expose plants to chemicals or radiation to introduce random mutations in their DNA, which can alter the expression of genes affecting root development. Once a mutant phenotype is identified, researchers use genetic mapping to locate the mutated gene on the chromosome. Further analysis of the clone gene can reveal its role in root development, such as influencing cell division, differentiation, or hormone signaling. Gene knockouts allow us to remove a specific gene and observe the effects on the plant, revealing the gene's function. Overexpressing a gene can enhance its activity, potentially uncovering its role in root development. Gene silencing techniques let us reduce the expression of a gene, providing insights into its function. Analyzing the phenotypes of plants with altered gene expression, such as changes in root morphology or growth, helps us understand the gene's role. By using these reverse genetics approaches, we can systematically explore gene function and uncover the genetic mechanisms underlying plant development. Arabidopsis, a small flowering plant, is a widely used model organism in plant biology research. Understanding the development and function of Arabidopsis roots has led to important applications in agriculture and environmental remediation. Insights from Arabidopsis root research have helped improve crop yields, nutrient uptake, and stress tolerance in agricultural crops. Arabidopsis is also used to study the effects of environmental pollutants on plants, which can aid in developing strategies for cleaning up contaminated sites. Arabidopsis is a powerful tool that allows scientists to gain fundamental knowledge that can be translated to real-world applications.